Hello everyone. So in this particular video we will be talking about special plant compounds which are phytoalexins, and the reference material which we are going to use, which I have used, is directly from the journals. That is, I have taken one review article as a reference as well as one research article for learning about phytoalexins. Okay, so let's begin. So the first paper which we are going to see is a review article which have been published in a journal of plant pathology, and the title of this review article is Harnessing Camalexin as a Sustainable and Eco-Friendly Strategy to Control Harmful Phytopathogens. Okay, so from the title they have used a phytoalexin, that is Camalexin. Okay, and they have mentioned in detail about this phytoalexin, and they have stated how this reported camalexin has different role in controlling several phytopathogens, including both bacteria, fungus, virus. Okay, they have included, they have documented every organism. Okay, so beginning with what are phytoalexins. So they have stated that phytoalexins are characterized as low molecular weight secondary plant metabolite and are synthesized in response to pathogenic invasion as a broad spectrum antimicrobial defense. So basically, these phytoalexins are secondary metabolites. They are low molecular weight, and these low molecular weight compounds are synthesized only upon pathogenic invasion. Once the pathogen is entered into the plant, a series of reaction is initiated, right? So alternatively, this compound is also synthesized, and these compounds are having a role of defending those phytopathogens. Okay, so in this case we will say about camalexin. So the source of camalexin are generally cruciferous plant. They have reported several plants which also include Arabidopsis thaliana, okay, which is a source of this phytoalexin. These phytoalexin has also been reported, or this compound contains a substituted thiazole, and this group is responsible for its antifungal nature. Okay, moving forward, it has been stated that in plants, the localized accumulation of camalexin in response to infection is restricted to diseased portions, while healthy sections do not exhibit such accumulation. They have stated that in a plant, when there is a localized infection at that particular spot, the synthesis of phytoalexin is initiated. Okay, apart from this, the healthy section do not exhibit such accumulation of phytoalexins. Thus, camalexin is produced by plants in response to infection rather than under normal physiological conditions. Okay. Then they have stated about the impact of camalexin on oomycetes as well as fungal phytopathogen. They have documented in a table, and to be very particular, they have stated different plant species. Then they have highlighted the concentration of camalexin which has been utilized in the in vitro experiment, the test system which is in vitro, and then the name of the organism. Name of the organism in the sense the pathogen which is responsible for infecting these host plants or these plant species. Okay. So majority of the plants which have been studied for the effect of camalexin in response to infection is Arabidopsis thaliana, and different effect has been stated here, like the camalexin has showed both resistance and susceptibility against phytopathogens, and similarly several effect has been reported. Okay, you can see I'm going to cite this paper in the section in the description box, so you can go through it and read as much as you want. Okay, so again this the table is continued, and you can see the variety or the diversity in the phytopathogens which are responsible for infecting Arabidopsis thaliana only. Okay, in the similar way they have documented about the bacteria, nematode, protists, insects as well as viruses. Okay, again they have documented a table, the plant species, then assay of concentration of camelexin, test system obviously in vitro, then name of the pathogen or pest. Okay, and then the effect they have studied. Majority of it are stating that the camelexin is showing enhanced resistance in the plant against the pathogen. Okay, or they are going to state that they are increasing the immune response, means they are simply resisting the pathogen. Okay, in simple sense. Moving ahead, the second part is they have added a image in which they have shown that generally the pathogen virus attack on the plant. Okay, then the production of phytoalexin is initiated. Also they have highlighted that this camalexin influences the microbiome associated with roots to promote plant fitness. This concept is stated below. Like first we are going to see over here. The first point is camalexin exhibits direct antimicrobial activity against several bacterial species, like we have seen the documented list of bacteria, like for example Pseudomonas syringae and Pseudomonas aeruginosa. But also it requires higher concentration of it in comparison to those in order to inhibit the fungal pathogen. Okay, so the inhibition is okay, but the concentration of phytoalexin varies in case of both bacteria as well as fungus. In the next point they have stated that the beneficial rhizobacteria means those bacterial microflora which are residing near the roots of the plant. For example, Pseudomonas fluorescens and Bacillus subtilis can induce systemic resistance in Arabidopsis by enhancing camalexin accumulation, particularly in response to necrotrophic pathogens. Necrotrophic pathogens are those which ultimately lead to death of the plant. Okay, so the relationship of camalexin with beneficial microbiota, in this section they have stated the interaction. They have mentioned that phytoalexins have been observed to influence these microbial communities in order to promote its plant growth. Within the rhizosphere, phytoalexin influences the microbiome, enhances plant fitness and growth. For instance, they have given an example that the release of coumarins from plant have demonstrated to enhance plant iron uptake by influencing the surrounding microbial population. In simple words, they are stating that there has been a synergistic interaction between the phytoalexins which are synthesized by the host plant upon infection as well as the microflora of the plant. Okay, in this case they have stated about the rhizospheric microflora, that both are synergistically interacting. One is acting as an antimicrobial defense compound, while the other, along with several plant growth promoting activity, also supports the phytoalexin biosynthesis. Okay, yes. Now moving ahead, another example have been stated, like for example fungus Trichoderma virens as well as Trichoderma atroviridae. Both are very famous fungal pathogens which are very well documented as a biocontrol agent or a growth formulation, plant growth formulation. Okay, they have been widely used in order to promote plant growth. Okay, there are several studies which have reported them. In this case they have stated that both of the fungus produce ISR, that is induced systemic resistance, that induces the accumulation of camalexin in Arabidopsis leaves, which offers resistance against Botrytis cinerea, which is a phytopathogen. Okay. Okay, so lastly they have stated about the application of camalexin in pathogen control. Like in present scenario, several formulations, both biocontrol agents as well as green formulations, have been utilized in field or in laboratories for studying the suppressive effect of those on the pathogen, right? So is it possible to study these phytoalexins also as a biocontrol agent directly into the nature? For example, they have stated that several in vitro studies have been carried out. Like we have seen, they have documented several bacterial infection which have been suppressed due to these camalexin, but all the studies have been reported here is in vitro condition. In vivo condition is also possible, but they have stated that significant field experiments are still in early stages of investigation. Means this is a new area of it can be said like it is a new area of research if one is thinking to utilize these phytoalexins, not only camalexin but also others. If someone wants to utilize these phytoalexins into the field, one can think about it that how a formulation or anything can be developed by using this as a substrate, right? Another thing they have stated that the approach, like when one is targeting to control a phytopathogen or a plant disease, the major approach is also focus, means usually focuses on the target specificity. If I am preparing a formulation, then I will usually target the phytopathogen specifically. Okay, it should not be like I have created a biocontrol formulation and it is suppressing all the microflora which is present over here that are influencing the plant growth. Okay, so negative impact should be less. So that's why they have stated that camalexin, though effective against various plant diseases, may unintentionally affect non-target organism. Okay, so study in that way is also possible. Okay, so they have concluded the paper lastly. Okay, so this was about phytoalexin, particularly about camalexin. It was a general overview. Okay, it is a short overview. The next paper I will give a short look into this. So this is a research article, okay, and it is published in Environmental Microbiology Journal. In this case, the title of the paper is Diterpenoid Phytoalexins Shape Rice Root Microbiomes and Their Associations with Root Parasitic Nematodes. In this paper, in this article, they have worked on diterpenoid phytoalexins, and the main focus was to study the interaction of these phytoalexins with the root microbiome during infection with a parasitic nematode. Okay, that is a root parasitic nematode. Okay, we will look into the abstract. Okay, so rice synthesizes diterpenoid phytoalexin, which is shortly given as DP. Okay, DP. These are diterpenoid phytoalexins which are synthesized by rice, and they are reported to defense against foliar microbial pathogen. Foliar microbial pathogens are those pathogens which infect or which target the aerial parts of the plant, that is the leaves or stems or flowers or fruits. Okay, and the root knot nematode, that is Meloidogen. Okay, so Meloidogen is a root knot nematode which they have used as a phytopathogen for the study. Okay, so they have studied the role of DP in shaping rice associated root microbiome in nematode infested field soil. What they have done is they have used 16S as well as IPS2 rRNA gene amplicon in order to study the root microbiome of two different plant. Okay, they have taken a wild type rice, and on the other hand they have taken a mutant rice which has DP knockout rice mutant. Okay, so these DP knockout rice mutants are not able to synthesize those diterpenoid phytoalexins. On the other hand, there is a wild type rice. Okay, and they have studied the microflora at 17 day, that is the early point, and at 28 day, that is the late stage. Okay, so they have observed that there has been a difference in both. There has been difference in both the knockout rice mutant field rice when sown in the nematode infested soil, the microflora is much varying than the wild type flora. Okay, they have shown that in case of nematode suppressive microbial taxa like Streptomyces and Enterobacter was negatively correlated with the this nematode. Okay, they are simply stating that the wild type rice has several beneficial microflora which were able to suppress the root knot nematode. On the other hand, the knockout mutant was unable to synthesize the phytoalexin. Along with it, there was deficit of such beneficial microbial taxa. Okay, so this is the major finding they have stated. These findings indicate a role for DP in plant microbiome assembly and nematode interaction, further underscoring the potential of leveraging phytoalexins for the sustainable management of crop diseases. So initially, like previously, we have seen that the first stage was like whenever a compound is studied, initially it is identified, then it is characterized, then its role in plant is reported. Like in this case, they have reported that they are involved in antimicrobial defense. Along with it, if we are using it for the management of crop disease, the second targeting approach or the second approach that should be highlighted or that should be studied is its impact in the beneficial microflora of the host plant in the field. Okay, so this was about phytoalexins. We have seen two examples. The first one was Camalexin and another one was Diterpenoid phytoalexins. Okay, so this was all about the today's video. It is a short video. I hope you guys like it, and if you do like it, please like, share and subscribe. Thank you so much.